This is Duke University.
So I just thought I would build up, like the drum roll building up to our big announcement, by just reminding you, this is a university of high achievers, and some high achievers are even higher achievers than other achievers. And sometimes they win prizes that indicate that they have achieved at the very highest level in which people like them compete. So I can remember when John Hope Franklin, our colleague, won the Presidential Medal of Freedom bestowed by President Clinton. John Hope Franklin is really the founder of the modern study of African American history and of American history as something that includes African American history. So that was a proud day for us. And then I can remember in the year 2008 when it seemed as if every single member of the Duke community might end up with a ribbon around their neck by the end of that year. And so I remember the Olympics that year when, you know, the United States has suffered shameful defeats with all its talents in previous Olympics because it only had good individual players. But then it took somebody to teach them how to become a great team, and that somebody was Mike Krzyzewski. And so after his team won the gold medal in Beijing, they put the coach doesn't get a medal, but they put all their medals around his neck. And so that made me very proud. And then very soon thereafter, we had an event in the Nasher Gallery in which our own colleague, Sarah Schroth, whose research was the foundation of the exhibit many of us saw called From El Greco to Velázquez, when she herself was, this is me, and this is the consul of the Spanish embassy who came here to bestow on her the highest order that the country of Spain bestows on any foreigner. She was made a Knight Commandant of the Order of Queen Isabel de Catolica, and she is hereafter entitled to call herself Su Illustrissima, or Most Illustrious One, which I think is a good title. Okay, so you had Mike, you had Sarah, and then that fall also another of our colleagues, Bob Lefkowitz, then won a pretty good medal. He won the National Medal of Science bestowed on him by the president. And so that was a memorable fall for me because you had somebody in art history, somebody in athletics, somebody in biomedical research, each wearing something around their neck similar to each other. And so for me, that gave me a sense of something very enduringly important to me, which is every achievement of excellence on this campus provides an image of the excellence we search for all across this campus. So it seems to me anybody's medal becomes a challenge and recognition of something we all strive for. This fall, a couple of our young pals won medals. You remember Abby Johnston and you remember Nick McCrory when they won the bronze and silver medal in diving at the Olympic. And so you thought, oh, this year the medals are going back to athletics, but once again it was a year in which we had athletic medals and then another medal. This medal has not been bestowed yet, and Bob, you for the first time are having to visualize what it will be like to be the Nobel laureate. But I want to say, my thought is this. Nobody gets the Nobel Prize by accident. There must be many worthy people who never get it, but nobody got it who didn't richly deserve it. And when they get it, it's a tribute to the individual, but it also brings reflected glory to the whole community of people who surround them. And I think your presence here today shows that you felt just that, the way in which Duke is honored or Duke is lifted up by the high achievement that Bob Lefkowitz has done. His work in biomedical science, he is the discoverer of the phenomenon of receptors, or shall I say the unfolder of the mystery of receptors. How do cells figure out what's outside of them? How does information come into the cell? When you see a scary thing and all of a sudden your heart starts racing, what was it that let each individual cell know to respond to the adrenaline that had been released? For this accomplishment, you won the Nobel Prize in chemistry, and what makes me almost as happy, somebody else shared the prize with you, but that person is not really other to Duke. He's your former Duke student who was here and was also a member of the faculty here for several years before you went on to this glory. So my thought was, in the last couple of years, I have had the pleasure of going around the country asking people of great accomplishment, of Duke faculty, administrators, alums, in front of large audiences and interviewing them, to squeeze them dry. What is it they know that we could all learn? So my thought was, why should I only do this in San Francisco or in Washington or or New York? Why not do it in our very hometown with our very own homeboy? Bob, you are our homeboy. You moved to Durham in 1973. You had already lived here for at least 21 years before people in this room were even conceived. You know that this is true. And so I say, if this community shouldn't hear something, I'm really glad that everybody in Sweden is going to know you so well, but why don't we begin by knowing you better here? So please join me in welcoming our great and distinguished colleague, Bob Lefkowitz. Now we're artfully putting these microphones on so that they face up, I hope. Yeah? Oh, no, let's try it this way. You're such a natural. You got yours already. Why don't you go ahead and start talking, and I'll ask you some questions. I'll ask you some questions later on. We can do it this way. Does that work? Does this sound okay? And Bob, are you?
Yeah, I guess I'm wired as well.
We're both wired. Okay, I know a story about you, which I heard a couple times the day you won the prize, which is that you sleep with earplugs in. And therefore, when you got the call from Stockholm, you never heard it. And no matter how many times they called you, you never would have heard the call. And maybe they would have said, oh, what the heck, we'll give it to somebody else. Your wife got the prize. So what I really want to ask you is, how has life been different pre-Nobel and post-Nobel? What's it like to be the new you?
Well, I think the new me is characterized by just a tremendous amount of activity, which I know will quiet down and go away. There's a lot of press attention. We have a TV crew with us today from Swedish TV, and they're with us today.
They're making a reality show based on you.
A reality show, exactly. We have another one in tomorrow. So it's extraordinarily hectic. I've been hearing from all manner of folks from different phases of my life, dating all the way back to elementary school.
Right.
And as you said, everybody is sort of lifted up by it and wants to touch it, and that's fine with me. I think the most interesting and, in a sense, amusing person who reached out to me was one that came by email. I received about 1,500 emails, congratulatory emails, in the first 48 or 72 hours. They continue at a much slower rate now. But this email was from an individual named Gary Lefkowitz. I do not know this individual, and he is no kin. But he wrote, and I'm quoting, "Dear Dr. Lefkowitz, congratulations, etc." He says, "I want you to know that all of us named Lefkowitz are standing a little taller today."
If I were Gary Lefkowitz, I would only ever call myself Professor Lefkowitz hereafter. This is good. Okay, well, if I see any tall Lefkowitzes, I'll know what the explanation is. Let me ask you a question a little more serious, which is, I got to speak at the public recognition when Coach K won the game that made him the winningest coach ever in the entire history of basketball. And you realize when you're approaching someone like that people who reach the highest level of accomplishment, they didn't just choose their career by random or go to the career center, that they've had a sense of calling, right? That there's something you were born to do or something you would only be fulfilled if you could do. But I also think, in my experience, often people have a sense of calling that, like, you woke up in the cradle and grabbed the microscope, giving early signs that you would win the Nobel Prize in chemistry. Tell us the true story of your calling and your sense of it.
Is actually that I was a very reluctant scientist. And I think for young people, there's some lessons in that. So I did experience what I would describe as a calling, but initially it was not to research at all, it was to clinical medicine. So beginning at a very young age, maybe third or fourth grade in elementary school, I formed the clear intention of becoming a practicing physician. I think this was based on a particular role model, and the role model was my family physician, Dr. Feibush in the Bronx, who made house calls. He was a general practitioner. And I was just enthralled by the whole concept that he would come to the house when I was sick, and he'd put this stethoscope on me. God knows what he was doing with it, but it just seemed like an amazing ritual, and I wanted to be able to do that. Mm-hmm. And so from there on out, I felt this calling to clinical medicine. I couldn't wait to get to medical school. I loved medical school, and I loved my early residency training. And I never envisaged a career for myself other than as a practicing physician. I favored internal medicine and cardiology. But then I had an experience at the National Institutes of Health in the late 60s where I was introduced to research. It was painful. The first year was an unmitigated failure. Nothing I tried worked. And I was not used to failure. In fact, I was used to a great deal of success. So this really set me back on my heels and convinced me further that my destiny lay, as I always imagined, in clinical medicine. But toward the end of my second year in research, things began to work, and I had my first taste of what it's like to have experiments actually work and maybe even discover something, albeit not necessarily something all that important. I then returned to clinical training full-time, but I really missed the laboratory. And gradually I began gravitating back towards lab work because I so missed it. So the other lesson is the absence of that was what really made my heart grow fonder.
Okay.
When I came to Duke,
have you ever made a house call in your life?
yes, as a matter of fact, I made many, but it was for a different reason. I held several moonlighting positions to support my growing family when I was a resident in Boston, and one of the 3 or 4 moonlighting jobs I had was to do insurance physicals for a company for $15 a shot. And I had to go to the homes of the individuals, so I would drive all over the place and do these physicals. I was eventually fired from the position, though, because they wanted me to play down any significant findings like heart murmurs, etc., so that the clients could get the insurance.
Shutting the career door to you as an insurance physician.
Yes, as an insurance physician, I didn't make it.
Well, but I like your story so far because you knew what you wanted to do. It's not that you had no idea. It's not that your career was undecided, but you know, how many 9 or 10-year-olds know what a research career would be like? So you found your way to the visible version of it, and then somehow you found your way, without being initially clear about it, to down a fork in the road.
This is true, and I think there's a very important lesson here. And it has to do with the fact that, you know, each of us is born with certain gifts and certain weaknesses. But, you know, you don't come with a little card, although that may happen eventually with the Genome Project, but currently you don't come with a little index card that tells you what they are. You have to discover these along the way.
Sorry.
And unless you are exposed to particular activities, you might never discover what you've got. And if it had not been for my research experience at the NIH, I would have gone into practice, and I suspect I would have been quite happy with it. But I would have missed all this.
But things went another way. So I actually have learned immense amounts in the last 2 weeks about the thing I call receptors, but that I'm told you call receptors.
That's the New York way to pronounce it.
That's right. So the discovery of the G protein receptor. I could give my explanation of your discovery, but it might actually be even more edifying to hear your account of it. Just what's a way to describe to an intelligent lay audience the thing you found?
Okay, so I think that the simple analogy that we use, and it's certainly not original to me, and it applies to other types of molecular interactions that occur, is the idea of a lock and a key. So hormones like adrenaline and neurotransmitters like dopamine, serotonin that we're all familiar with are in a sense keys. They need to fit into the cell, change something, and start a process, a cascade of events that will change what's going on in a cell and thereby in an organ. Let's take the specific example of adrenaline, the fight or flight reaction. When you are excited or nervous or stressed, you secrete adrenaline from your adrenal glands. It circulates in the blood, and it will ultimately make your heart beats stronger and faster, and it'll prepare you in a variety of other ways, which I won't go into for prominent activity. But how does it know to do that? I mean, how does it know to work on the heart rather than a cell in your nose that smells? Well, the answer is there are these specific recognition sites, which are the locks, and they have a complementarity in their structure to the key. Now, in the case of a hormone like adrenaline, it's what we call an agonist. That means it stimulates something. So it fits into the lock. The lock is an adrenergic from adrenaline, adrenergic receptor, and in particular, the one in the heart is referred to as a beta adrenergic receptor. It fits in and it turns the lock. Okay. If it didn't fit exactly, it wouldn't get in there in the first place. But it more than just fits, it's able to do something. It's able to activate what we call cellular signaling and start a cascade of events that leads to the heart beating stronger. In terms of therapeutics, what's very important is that there are molecules that can stick into the same receptor, but they can't turn. It's as if you had a key that was broken off. They're called blockers or antagonists. And many of you have probably heard of beta blockers, which are used in cardiovascular medicine all the time. They bind to these receptors and they block the effect of adrenaline. There are similar examples for a host of these receptors. Okay. One that probably most in the audience are familiar with are antihistamines, would be a classic example. They're antagonists. They fit in and they block histamine. You don't want what histamine is doing. In patients with cardiovascular illness, we don't want often what adrenaline is doing. So that's the whole idea. And my whole research career has been involved in first proving that there were such things.
Well, okay, because let's go back to that moment. I'm told, that, or at least it was described to me by a colleague of yours, that at the time you came into the field, it was hypothesized that there must be such a thing as receptors, or such a mechanism as you have just been described. But this had been deduced more than it had been observed.
This is correct. This is correct. Nobody had ever proved that they existed. And in fact, while much data from pharmacologists was compatible with the idea, there was tremendous skepticism as to whether they even existed. And in fact, when I lecture on this topic, and I'm just putting together my slides for the Nobel lecture next month in Stockholm, I will present several quotations from prominent pharmacologists of the 20th century, going right up to the mid-70s, expressing total skepticism as to whether the whole idea of receptors was even a useful one, much less that they might exist as actual physical entities.
So then, a person like me who has not had a career as a research scientist, the question would then be, so someone has hypothesized that these things exist, but it hasn't yet been demonstrated. How do you find it? It's not like you look under the bed.
Right. So much of science is driven by the development of new technologies.
Okay.
And if you're going to make really game-changing discoveries, more often than not, you're going to have to invent technologies that don't exist at the time. And when I first came to Duke, it was clear to me, this was in '73, that if we were going to have any chance of succeeding at what I wanted to do, which was to prove the receptors existed and then move on to find out what they were all about, we would have to develop techniques that didn't exist. And the main one was some way of seeing that receptor, of tagging it. And the idea was about, at that time, that you could use radioactively labeled molecules. For example, a radioactively labeled adrenaline molecule or a radioactively labeled beta blocker molecule might bind to the receptors, but nobody had been able to really do it. And so that was the first project that I set out to accomplish. And with the help of my first remarkable MD PhD student, Rusty Williams, who went on to a pretty good career of his own, elected to the National Academy, on our board of trustees here at Duke in the past, and one of my first postdoctoral fellows, Mark Carone, who's still at the institution as a James B. Duke Professor of Cell Biology. We were able to crack that problem within a year, and everything kind of flowed from that.
Well, so tell me, Is there some moment of discovery, or is it the accumulation of data where after a while something comes clear? Or was there ever a moment when you said, Eureka?
The answer is yes, 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 and yes. I mean, there are a number.
I'm so glad you don't disappoint me.
There are a number of Eureka moments, some more Eureka than others. But there's also the accumulation, one thing building on another. If I had to choose a single Eureka moment that, and there have been a number, I've been very fortunate, the one that stands out in my mind is one from 1986, which was actually made together with Brian Kobilka, the individual who was a postdoc in my lab, a cardiology fellow at the time, and who is now the co-recipient of this award, and who's a professor at Stanford. We had just succeeded in cloning the gene with the so-called beta adrenergic receptor. I had spent, excuse me, about a decade isolating the receptor and characterizing it, and that gave us the tools that we needed to now clone the gene and learn the entire amino acid sequence of the protein. And when we did that, we could see a particular organization to the protein where the protein chain weaved across the plasma membrane, the cell membrane, 7 times, okay? Sort of like a snake. There was only one molecule known at the time which did that, at least one known in animal cells, and that's something called rhodopsin, which is found in the retina and is the basis for how we see. It is, in a sense, a light receptor. Photons of light impinge on rhodopsin and change its conformation or its shape in a way that lets it do something which is kind of analogous to what a hormone receptor does. It activates a G protein and it leads to a cascade of signaling, which in that case leads to visual processes. So in that moment, when we saw not only that it shared this arrangement with rhodopsin, but that some of its amino acids were exactly the same as rhodopsin, it occurred to us, my goodness, maybe all of these receptors are going to look like that.
Right.
Okay. And in fact, within the next year or 2 or 3, we were able to clone the genes for a number of others. They all did look like that, and that really opened everything up. That was a true Eureka moment.
Well, you saw something, or you realized something with your mind that no one had ever been able to make an object of consciousness or an object of knowledge before.
And this experience is remarkably seductive.
It must release hormones.
Yeah, it does indeed. Adrenaline amongst others. But you know, one of the things I like to do with my students and fellows, especially the ones that don't have any research experience, is to try anything I can to get them to the point where they can make even the smallest of discoveries. And you know, in a sense, any experiment that works and gives a positive result is a discovery. Might not win you the Nobel Prize, but it is in fact a discovery. And once you experience the exhilaration of knowing that you know something in that moment that nobody else knew, as I say, even if it's very small in its import, it's very seductive.
It's amazing. It's amazing. Well, let me ask you then to broaden this a little bit about scientific inquiry more general. I was at a meeting in New York last week and someone was introduced, also a distinguished scientist, and the story was told that many people's parents would say to them when they came home from school, "Did you get good grades in school today?" But that this person's parents had said, did you ask a good question in school today?" And I'm going to guess that this has something to do with, you know, that good scientists must be people who work on good questions.
Well, I think this is exactly right, and there are 2 types of questions. The big question, okay? Do receptors exist? That would be a big question. But then, as important is the specific question. By that I mean the experiment you're doing today. So every experiment is a question, okay? But just like I suspect, and I have no experience in this realm, just like a really good cross-examining attorney, if you don't ask exactly the right question, you may not get the most meaningful answer. I'm aware of, sometimes I've read about cases where somebody, has committed perjury, but they managed to get off because nobody asked them exactly the right question, and so they were totally honest. Well, same thing in what we do. And each time you ask the question, that is, do the experiment, you get a little sharper. So you may do it the first time, and you get the answer, and you say, well, that's okay, but I didn't do that experiment right. So now you sharpen the focus a little bit, and again and again. And when you do just the right experiment, then you get the answer that'll take you to the next level.
But in your account, you don't start by getting the right question. You start by asking a question and doing something that helps you figure out how to sharpen the question.
Right, you start by, everything is determined by asking the right big question. Because if you're asking the wrong big question, none of the other stuff matters, and everything you do will be trivial.
Okay. Well, I'm interested, you talked about how anybody who's ever had the experience of discovering anything, however small, will have felt the intoxication or addiction of that. But there must be lots of people who go to labs every day who don't discover anything that day. So could I ask you about failure in science? Science
is 99% failure and 1% success. Now, that's an optimistic view, okay? Most of what we do is fail. And it's a very difficult part of the profession. I know in my own case, it almost drove me out. After my first year at the NIH, I had met with no success at all. And as I mentioned, I was not used to failure. And it was very hard to take. It was at that point, since it was only a 2-year hitch, if you will, in that position, it was at that point, after 1 year, that I made arrangements to move on at the end of the second year to more clinical training. Now, by the time I finished my 2 years, I really would have preferred to stay because things were going well. Right. But I hadn't learned to deal with the frustration yet. And I thought, if I can't do this in a year, I just don't have it. Now, again, if you are asking big, important questions, you do have to be prepared to fail. And that's one of the most difficult things to help people to come to grips with.
Well, Howard Ruckman's in this audience, and he told me that you told him early in his career that if his experiments were successful 80% of the time, it meant he was doing junk science?
Well, not quite maybe that stark, but I actually go through, some of my fellows are in the audience here today, and maybe they shouldn't hear this, but at the end of every year, in the privacy of my own office or home, usually during Christmas week, things are very quiet, I review everything we're doing. And if we're succeeding at too many things, I'm not happy, because it means we're not really asking the really important questions. And I think many of them in the audience will agree with me, we're asking the right questions right now, because we have a lot of failure. But we're getting there.
Well, I mean, it's incredibly interesting, and I think that people who don't do this for a living don't understand this about the nature of inquiry, that the failures are a means to advancement, but also they're just an inevitable consequence of the lack of knowledge in the first place.
Absolutely. Every failure makes you smarter. You know something that doesn't work, and so you can move on to the things that do. One of the interesting and, in a sense, unfortunate parts of the scientific enterprise is that you pretty much can't publish what are called negative data. That is, things that didn't work. Only positive data. Yeah, the problem with that is it means others are doomed to repeat your failures because they don't know it's a failure, because you don't write about that. But
so think how long our vitae could be if we could publish all our failed experiments.
Oh my goodness.
Yeah. So let me ask you another question. We speak at this university, we often speak about knowledge in service to society, and you certainly come to my mind in that regard because you made discoveries in the domain of knowledge that have been of service in helping the treatment of heart disease, helping the treatment of schizophrenia, helping the treatment of the common cold. And didn't someone tell me that half the commercial medications on sale in this country work by working the receptor sites you discovered? So that's service in a big way. And actually, I love the fact I have studied the Latin that's what's on the Nobel medal you're going to get. It's a line from Virgil, and it's an amazing line about those who have bettered human life through their discoveries of new arts. And so that's what you've done. And so it's a betterment that came through a discovery. But an interesting thing to me as I hear you is, I don't hear you saying, I got up in the morning saying, today I'm going to find the cure for schizophrenia. Instead, you said, today I'm going to find something about the G protein, something of that sort. So I'm trying to ask you a question, which is, to what extent does the long end benefit of your work function as a daily motivator of your work, or is it just the happy consequence, the happy byproduct?
Yeah, that's a wonderful question. You know, there are various types of biomedical research that go on. Often people will identify, I'm thinking of three types. At one end of the spectrum, you would have what I call real clinical research, by which I mean you could shake hands with your experimental object. Okay?
Sure.
We have a wonderful clinical trials unit here, the Duke Clinical Research Institute, one of the leading clinical trials
in the world.
Enterprises in the world. So that would be real clinical research. You know, every day when you come in, you're testing whether a drug works in human beings. It doesn't get more on the front line than that.
That's right.
Then at the other end is kind of what most of my research is. It's basic fundamental research. Do the receptors exist? What are they? How do they function? How is it regulated? How can we manipulate them? And then in the middle you have what's called translational research, which is generally taken to mean trying to apply basic findings, maybe not necessarily right at the patient level, but in animal models. I have a component of such translational research, but throughout my career, most of the research has been basic and not necessarily motivated by what disease am I going to cure, or the specific focus to cure a disease. That said, as a physician investigator, everything I've ever done is informed by the fact that I was and always will be a physician. And so anytime we've made an advance, as we like to say, it does not escape my notice that there are particular implications in terms of either a disease or drug development, this kind of thing.
So this is really interesting because the way you phrased your answer, it's not a real bifurcation of basic and applied or basic and translation, because it was your original care that actually led to your work. But at the same time, you know, you can't solve schizophrenia by wanting to solve schizophrenia. I mean, if you can't find the underlying mechanism, if you can't, at the level of pure research, make that discovery, then the therapy isn't going to
Exactly. In other words, let's say you set out to cure schizophrenia. Okay, so then one is, okay, now what? You have to be studying some basic aspect. And in fact, the discovery that cures schizophrenia, to use that, will probably come from somebody who's not even thinking about that, but just he discovers a process and then somebody else notices there's a relation here, and then that's how it's going to happen.
Okay. So one thing I hope people in this audience are aware of is, in addition to your discovery of receptors, this is also one of the really famous teachers in the world of biomedicine, but also in the world, I think I could say. And I love the fact that, you know, I know you have trained something like 200 or 220 fellows who've gone on to important careers of their own. And I love the fact that you shared the award, not in the sense that the honor was divided in half and you only got half the sandwich, but in the sense that you were honored for what you did and for your work helping someone else go on to his own discoveries. So let me ask you some questions about teaching, okay? Okay. What makes a good student? Well, you get to pick yours. You know, when you admit people to college, that's one thing, but when you admit somebody to your lab, that's a little different. What do you look for?
Well, I think in general, what makes a good student is curiosity and interest.
Hard to get far without them.
Those would be the two major things. Now, what I'm looking for when I select people from my own laboratory are some additional factors. One is a real work ethic, and another is a sense of optimism. And again, unfortunately, these things are not easy to evaluate in the very short time frame that I have available, typically a day when they come for a visit and some letters. What more do you have?
You came over to my house the year
Yeah.
you won the National Medal in Science,
Yeah.
and we had a fun chat that I still vividly remember. And you told me that when somebody comes to interview for your lab, you ask them a question, and it's a question I have never ever asked a student. You told me you asked them, "Are you lucky?"
Yeah, I do ask that question. Not all the time, not all the time, but maybe half
What
the
was
time.
that? What was that about?
Well, what it's about is I'm a big believer in self-fulfilling prophecies. And of course, there is such a thing as luck or fate or destiny. You cross the street and you get hit by a car. I mean, that's bad luck, assuming.
But nobody ever crossed the street and found the G receptor.
Well, exactly. So you need to, for the most part, make your own luck. And to me, luck is sort of a matter of perception. And I really do believe that people who believe that they're lucky are probably going to wind up being luckier than those who believe they're not lucky. And I can tell you, when I do ask that question, if somebody's answer runs to, well, not really, it's always kind of raining on my parade and this and that, you can be sure they're not coming to the lab.
Note to self. Okay, so I asked you what you look for in a student, and let me now ask you the complementary, the question that fits that lock. What can a teacher do for a student in your?
Say again?
What can a teacher do for a student in your world?
To me, the single most important thing that let me use the word mentor, if I can, because anyway, I think that applies more to what I do, because I don't give a lot of formal lectures.
I understand.
This and that. But to me, the single most important thing that a mentor can do, and you don't do this in one session, is help the student find out what their true gifts are and to somehow enable the student to tap into that and learn to play to that strength, while at the same time shying away or trying to minimize the adverse consequences of the deficiencies we all have. Because when you have done science as long as I have, you learn that there isn't one right way to do it. I mean, you couldn't have two more different personalities than Brian Kobilka and myself in every respect. But we've both done okay. And the reason is we do science a bit differently, but we each play to our strengths, and we each know what our strengths are. And you may or may not get to the point where you can actually articulate them, but that doesn't matter. As long as you intuitively have a sense of who you are, then you'll do okay.
Well, I've thought of you as illustrating the two core activities of a university, right? One is inquiry, inquiry-driven research, and the other is teaching. And I understand teaching meaning not to give lectures, but rather to help people discover themselves, to help people release their powers to their own careers. And so that's pretty much what you're talking about.
That's exactly it.
So let me ask you, you must have found Duke a good place to do these things. Either that or you're a profoundly immobile person, because you came here 39 years ago, right? And you unpacked, and you never left. Tell us a little bit about how you would characterize Duke as an environment.
Okay. So first of all, the university was a fine university when I came in '73, but it was not the powerhouse that it is today. When I came to Duke in 1973, from Harvard, as we like to say in Boston, I never dreamed I would spend the entirety of my career here. I thought that this was an up-and-coming university. I liked the tuition benefits, since I already had 4 kids. But yeah, I really didn't think I was going to stay all that long. I hoped maybe if I could have a good 5, 8, maybe even 10-year run, then I'd return to some place like Harvard or Princeton or Columbia, my alma mater. But I just found the environment wonderful. First of all, I liked the physical environment. I mean, I liked living in an area which didn't have the distractions and frustrations of a city like New York, where I grew up and where I love to visit, but frankly, I wouldn't want to live. So I really liked raising my family here. But I found the place very collaborative, very comfortable. It was very supportive of me. And I just stayed on and on and on. One of my life principles has always been, if it isn't broken, don't fix it. And God, it seemed to be working.
Perfect.
And I was very happy, as I am now. And here I am 39 years later.
Well, now let's take you forward through the time machine and say you were leaving medical school right now and thinking of a career of biomedical research. I've heard people say that this remains the biggest area of discovery that will ever be open in our lifetime. And I've also heard people say that due to funding things and various mechanisms, the career of a biomedical researcher has gotten significantly more challenging to start in. And I think you and I are both aware of this thing like the ARISE report that Nancy Andrews participated in, documenting that the average age of people who get their first independent grant in biomedical research is like 40 years old or something like that.
That's correct.
Just give me your assessment of is it an illusion that things were easier in the palmy days when you got out? Is the world always hard, except for people who have the insistence on going forward through it? Can you generalize in any way about that?
You know, I think that there's a fair bit of truth in what you've just said. When I think back, there are certain objective ways to measure this. For example, the NIH in the current environment is able to fund, I don't know what the pay line is now, of all approved grants, maybe it's something like 15%. In some institutes, it's even lower. When I started out, the pay line was like 30%. That's a huge difference in terms of right. What it takes to get funded. Right. So that's a big impediment. It is harder these days, there's no doubt about it, to get the independent funding. That said, the very best people do, of course, rise to the top and are generally successful. But we need, as a society, to understand how important the biomedical research enterprise is to our societal well-being. And we stand on the cusp, as you mentioned, of discoveries in medicine, which can be life-changing for so many individuals. But if we don't put the funds there, we won't be able to sort of realize that in as timely a way. I've been remarkably blessed in my career to have had support from the Howard Hughes Medical Institute since 1976. I'm the longest serving investigator that they've ever had. Mm-hmm. And I would point out that's a competitive process, so I've had to sing for my supper every 5 years the entire time.
I knew that already.
Yes, so there are agencies or foundations like institutes like the Howard Hughes Medical Institute which help, but alas, they have 300 investigators.
Well, let me ask you one last question, and then we'll take a couple from the room. And I don't want you to think that there's any hidden rudeness in this question at all. Okay, I'm able to calculate that you must be, say, somewhere not too far from the age of 70. Is that right?
About 6 months.
Okay.
Plus or minus.
Yeah. Orders of magnitude, right? At the same time, having known you from the first time I met you, you could the inner you is so visible from the outer you, and the inner you is somebody who's like a kid. Is this right? Okay.
You've hit it right on the head.
So do you see yourself retiring? Do you see yourself having a post? Do you see the Nobel as being the thing that might just, you collect the Nobel and get my handshake and...
So I would have to say...
Or do you see yourself never retiring?
I would have to say that the latter, but of course I'm realistic enough that things sometimes often out of our hands as we get older in terms of health. But let's say for the moment that that doesn't become an issue. It's hard for me to imagine retiring. I'm impressed that you really nailed me. I'm basically a kid in an aging body. I have used the...
Your body's not
Not totally
Not decrepit
decrepit. It's not totally decrepit. It still can last an hour on the treadmill every morning. But that aside, I have often told people in my career when the subject has come up for discussion that I can honestly say that, except for the rare point in time when something is going on, that I've never viewed myself as working for a living. I mean, I've never had a job, and I don't work. What I do is my sandbox. I come to work every day, and I play. And I play with the students and the fellows, and anybody who works with me knows that I have an immense amount of fun. And then I go home, and I enjoy my time at home. A good part of that is spent reading stuff related to
the sandbox.
What I do. But the sandbox is, to me, is sort of everywhere. And it truly is play. I mean, I don't think I have ever viewed myself as working for a living, and I don't now.
Well, lucky us to have lived somewhere near the sandbox of Bob Lefkowitz. Can I ask if we don't have infinite time, because actually we had to promise we'd be out of this room in time for a 5:15 event. But let's take some questions, and there's microphones on both sides. Here's one over here. If you wait till the mic comes, we'll all get to hear it. Tyler will run as fast as possible. Right here. Oh, you don't have the mic. This guy right here. Sure.
Hello?
Yeah.
So Professor Lefkowitz, congratulations on winning the 2012 Nobel Prize in Chemistry. And my question would be, you said that 99% of the time you experience failure instead of success during your research. And so how do you actually deal with that large amount of failures? Like, do you try to persuade yourself in, continuing your research? I mean, that must be really, really hard for you to continue your research for so many years. Thank you.
Could you rephrase? I had a little difficulty.
The question was about what it's like if you say 99% of the most successful career is failure. How do you motivate yourself forward through the failure? I think it's actually a great question.
Yeah, I think as with everything in science and life, you have to find your own way. That is to say, not everyone will deal with it in the same way. For me, I managed to focus on what is working when I get sort of down about what isn't working. It's a lot easier for me, not only now, but for many years, in that I have, a group of people working. And at a time when, say, several people are having problems or are hitting roadblocks, of course, I have access to other individuals in the group who are succeeding. Of course, that doesn't help you when you're at your stage of the game. And I think one has to develop certain calluses, and this only comes with life experience, in terms of how to deal with those difficulties. But I think each one has to sort of, live through the I think the more you go through failure and you come back and you see, you come back on the other side and you have that experience, the more helpful that is. And you need to just try to remain optimistic.
Going once?
Yeah,
Going twice? There's one way
yeah.
up there. Oh, here's one. Okay. And Jordan, there's one way up at the top. Please.
Dr. Lefkowitz, what exactly does winning the Nobel Prize mean to you in terms of what your contribution has been to science?
So the question is, what does the Nobel Prize mean to me? Well, I must say, it gives me a tremendous sense of sort of inner satisfaction. I think I probably respond differently to it at 70 than I would have at 40. I've had a long career. Hope to have a lot more ahead. But yeah, it is a validation of not just what I've done, but of what my whole group has done. I wish in some way they could give awards like this to the whole group, because so many people contributed along the way, and that's not unusual at all. I'll tell you, and I say this in all honesty, and it's not just because the president is sitting here, but the fact that I was able to bring this to Duke gives me an extraordinary sense of satisfaction. I've been a Dookie for 39 years. 2 of my 5 children graduated from Duke, undergrad. I've had seasons basketball tickets since 1976. So I'm a Dookie through and through. And on the day the prize was announced, through many interactions that I had, both with individuals, at the press conference, and then at a lovely reception afterwards, and many times since, I sense this very strong sense of institutional pride, sort of like Gary Lefkowitz. And, that just makes me feel good. It really does.
That's great. There was one up here. Thank you.
I have a question regarding your early discovery about receptors. And my question is really about how much pushback did you get? You talked about it. You were saying that you had a lot of quotes in some of your presentations. Can you talk more about some of the pushback regarding your discoveries?
We both missed one word you said. Could you talk some about and then what's the next word with regard to your discoveries?
Some of the,
Pushback,
like contention
pushback.
And...
Okay.
Pushback that I had.
In the days before everybody agreed that you'd found it, when there were people who doubted that there was such a thing.
Right. So there was pushback at several stages. I'm glad you asked that question. When we first developed these ligand binding methods, these radioactive tracer methods, many classical pharmacologists were unhappy with this. And one of the slides that I like to show is a quote from a scientist named Raymond Ahlquist. So Raymond Ahlquist was a very famous and distinguished classical pharmacologist of the mid-20th century. He won the Lasker Prize, a very distinguished prize for biomedical research. He won that prize for publishing a paper in 1948 in which he used classical pharmacological approaches and took about half a dozen molecules similar to adrenaline, say congeners of adrenaline, and looked at their ability to stimulate various physiological processes, such as making the heart beat faster, making a blood vessel constrict, making some glandular tissue secrete its fluid. And based on several different patterns that he observed, he postulated there must be 2 types of receptor for adrenaline, which he called alpha and beta. In 1973, shortly after I came to Duke, and I was talking about, before we even were successful, I was talking about our attempts to develop these initial methods. He and I served on a panel. He was an elder statesman. I was a brash young 30-year-old. And he wrote, published, something called Perspectives in Biology and Medicine. He wrote, I don't remember the exact quote, but it was basically that he didn't believe in any of it. To him, he used the term receptor, but it was some vague pattern of forces. And he said there were actually some people, and these weren't his exact words, I'm paraphrasing, there are some people who actually had the chutzpah, not his term, to actually think these things exist and that they can study them and isolate them and find out what they are. He thought this was an act of sheer arrogance. So that was an example of the pushback. Another kind of pushback that you get, and I think this is probably true in many walks of life and certainly many areas of scholarship, is if you don't come from the right pedigree. So there was a Nobel Prize awarded in the early 70s to a scientist who had, named Earl Sutherland, who had discovered cyclic AMP that some of you who take biology may have heard of, the so-called second messenger. And his descendants were, by which I mean people in his lineage, were amongst the most prominent young scientists working at the time. When I met with my first successors, I was really kind of an outsider. And when I would go to conferences, I wasn't one of the boys or girls. There weren't many girls back then. I wasn't one of the boys. And so there was pushback just, if you're coming into a new field from outside, you're not necessarily accepted so quickly. Although the fact of the matter is, if you look at the history of science, important discoveries and changes often come from outside a field because your thinking and approaches get kind of ossified. So there was that kind of pushback as well.
Okay. But you push back on the pushback. I see a mic.
before going into research helped you versus having gone to do a PhD alone or done a MD PhD first?
Yeah, it's a very interesting question. And tell me if I got it right. How do you feel about doing a residency first? That's what you said, rather than doing a...
How did it help you focus what you decided you wanted to research in the long term before you started researching?
The question is, what's the best path?
Yeah, what difference do the different paths make? I mean, I've heard many people comment about you that you won the Nobel Prize in a field that you actually are not recognized as a practitioner in by everybody in that field, right?
I'm not recognized as a chemist, although I wish to share two recent things about that. Within the last 48 hours, the following two events have occurred.
He's been discovered to be a chemist.
Yes. One, the chair of our chemistry department has written to me inviting me to join the department. And I have responded that I would on the condition I didn't have to attend the faculty meetings. And the second, true story, day before yesterday or yesterday, I don't recall, I received in the mail a letter from the CEO and director of the American Chemical Society, which is the preeminent professional society of academic and, I guess, commercial chemists. I received a letter, one, inviting me, please, begging, to join the American Chemical Society. And two, no joke, including a personal check from the CEO of the American Chemical Society, made out to the Chemical Society for the $150 fee for the first year's dues.
You mean that you were going to get to pay it?
You're right. So I put that in the envelope, or my secretary did, together with she hadn't filled out the form for me, and I hate forms, but we did that, and we included the check, and presumably I will be a member of the ACS very shortly.
It's so disappointing you're becoming reputable at last.
Indeed. But,
okay.
But to the question about the best route, in my day there were no MD PhD programs.
That's right.
And there was this amazing selection process, filtering process at the NIH, largely driven by, I think, the desire of physicians for whom there was a draft, a conscription at the time, to avoid service in the Vietnam theater. This was an alternate form of military service. And so the best and the brightest, so to speak, competed for a small number of positions. We were all physicians. None of us had any formal research training. Remarkably, and I merely mean remarkably, in my group of 8 or 10 who served from 68 to 70, all physicians, 5 of us now have won the Nobel Prize. So first point, you don't need to have a PhD to do very good basic research. That said, I'm a big believer in our MD PhD programs, and that's been an evolution in my thinking. But there is just so much to learn these days that I really do think an MD PhD program is probably the best bet for a career in basic science for a physician. That said, can you do it today without that? Absolutely. Kobilka in the 80s, like me, was a cardiology fellow at the time, has gone on to do amazing things. You don't need to have a PhD. The important thing is you need to be smart enough and have the drive and the curiosity and the focus, the almost obsessive focus on an important problem. If you have those, I think the particular route you take is almost immaterial.
I'm going to take one more, and I think that the mic has been given up in this area.
Hello there. Congratulations. So earlier you talked about the sense of exhilaration that you got from perhaps being the only person that knows something. Could you explore that idea a bit, and specifically, do you ever feel that that became a polarizing experience for you?
You help me with it.
Could the experience of discovery be, do I have the right word, a polarizing experience?
That sense of exhilaration that you get from knowing something that isn't common knowledge.
Okay, but I'm still groping for the question in the question. The moment when you know something, I mean, presumably you never desire to hoard it. You're going to share the knowledge. But so give me another synonym for the word polarizing in that question.
Did you ever feel disconnected from people because you couldn't really express what you were feeling?
Okay, see, polarizing in that sense, that's to say that you're sort of lifted up. It's like in a state of exaltation where you feel disconnected. Well, come on.
No, I don't smoke that stuff.
Aw, you know what he meant. You know what he meant. Listen, I'm promised to get you all out of this room by a certain hour, and since I don't wish to get any parking tickets or anything like that, I'm going to do it. But let me just say, it has been my great pleasure to ask such a communicative person, and on this day, we are all Gary Lefkowitzes. Here's to you, Bob. Thank you.
Thank you.
Produced by Duke University. Online at duke.edu.